All right, and Aby, if you're okay with it, I'm going to do just a quick introduction before we let you loose.
That's good.
All right, so it's my pleasure today to introduce Aby Joseph, who's pursuing his PhD in optics. And Aby joined the University of Rochester optics program after a bachelor's degree in engineering physics from the Indian Institute of Technology, Delhi. And it wasn't long after he joined the University of Rochester group that he took a keen interest in imaging science and how imaging the eye could lead to impactful work in the field of optics. He joined the Advanced Retinal Imaging Alliance group under advisement of David Williams and myself. And you'll see that in Aby's thesis work, he really tackles a diverse array of imaging strategies of mammalian biology that has a single common thread. And that thread is focusing on the fourth dimension of imaging systems with an emphasis on time. So with time, we can reveal the beauty of biology, and we usually call that function. And to achieve this, Aby has developed an impressive temporal bandwidth where he can reveal the details and function of objects moving exceptionally fast, as well as those that move at glacial pace. And to give you a quick example of this, we in imaging science recognize that if you have some of the best optical systems in the world, but you have slow exposure time or long exposure time, you get something called motion blur, which wreaks havoc on the fine detail that you get in a biological image. As you can see in this image from a lion's mane being blurred by the shaking motion during the camera capture time. So Aby's work has combined high resolution that we get with adaptive optics imaging for the eye to reveal the frozen motion of things that are moving very fast in the back of the eye, such as moving blood cells. But we also recognize that time can be problematic on the other side of the temporal spectrum as well. If we had a camera that only took snapshots in time, we might miss the very elegant and beautiful dynamics of biology and life in full bloom. And for this, you might take a high resolution image of something like seeds growing, but you might miss the great detail of the growth of those seeds over time. And with this, Aby has applied incredible rigor and persistence in imaging things that are happening over long epochs in time and being able to see, for example, the remarkable immune cell dynamics inside the living eye. Aby has a long list of accomplishments in his time here at the University of Rochester, highlighted by 3 peer-reviewed publications, 2 of which were first author in the high-impact journal eLife. He has 2 other publications that are either submitted or in preparation. And because Aby is not only talented in academic writing, he also has a number of awards, including the Best Poster Award at the U of R Neuroscience Retreat, the Leica Imaging Award, which remarkably was given to someone in the field of optics in an immunology-heavy conference. He's been an invited speaker at a number of different events, including those hosted by the OSA. And over the past 2 years, he's even been a podcast guest at the BBC-produced Naked Scientist podcast that was produced 2 years ago. He's not only been very good in the academic realm, but also taken what he's learned in academics and applied this in IP, and has 2 submitted patents, one which has been issued and a second that we're waiting a decision on shortly. Without further ado, I'm going to hand over the torch to Aby. The title of his thesis, appropriately, is Imaging Single Cell Dynamics in the Living Eye from Milliseconds to Months. Or those of you that appreciate the growth of an individual, you might consider an alternate title. And that title is What Happens When You Plant a Good Seed, Give It a Little Soil, Water, and Air, and Watch It Grow. Aby, the floor is yours.
Thanks so much, Jesse. Thank you. Yeah, it's a pleasure to present this thesis. Jesse, just want to confirm that you're seeing my main slide and not the presenter view?
Yes, Aby.
Okay. Great. All right. So without further ado, I'll dive right in. And thank you so much, Jesse, for the introduction. imaging single cell dynamics in the living eye from milliseconds to months. When studying biological systems, specifically the central nervous system, the study is performed at multiple levels, starting from the whole body over the central nervous system, down to maps and networks, down to individual cells or neurons, further down to synapses and molecules. When looking at the many techniques which help enable the study, it helps to look at the techniques on a spatiotemporal plot, like the one shown on the right here. The plot is over space and time, and both axes are in log units. Over the space axes, you can arrange techniques based on a window where the lower end determines the spatial resolution of the technique, and the upper end of the box determines or shows the extent of the technique. So in optical imaging, for example, you can think of this as the field of view of the technique. Similarly, along time, you can define the range of a technique based on the resolution on the left and the extent of time on the right. And it helps to think about this plot as we go forward into this thesis. As Jesse just beautifully outlined, this thesis is conceptually organized on a time axis where seconds is displayed in log units. We're going to go from hundreds of microseconds to seconds to hours to weeks to months. The first part involves imaging vascular cells in health from milliseconds to minutes. The second part involves imaging immune cells in health from seconds to months. And finally, we're going to apply these techniques in imaging vascular and immune cells in disease over the entire temporal bandwidth. Now, when we have to study the brain in cellular detail, one of the things that makes it very difficult is that the brain is encased in the skull, and especially optical imaging techniques face the challenge of very high scattering and attenuation, which prevents us from studying the brain at a cellular scale. One way to circumvent that is to use the eye as a window to the brain. The light-sensitive part of the eye, the retina, is a part of the central nervous system with many cells and organization which are similar to that found in the brain. And as you can well imagine, the eye is optically transparent, at least at the wavelengths that we're interested in. And this gives us an unperturbed view of the retina through these transparent anterior optics. In this thesis, we are interested in imaging the retina in vivo in living mice and human beings, and imaging the retina at a cellular scale. However, there are some further challenges that need to be overcome for this. Even though the eye is transparent in the front, as we zoom in to get a view of the retina using commercial or conventional systems or ophthalmoscopes, we can get a very nice view of the large scale of the retina, including the optic disc, as is shown here, and the large blood vessels in the retina, and also differences in pigmentation at a global scale. However, when you zoom in further to smaller and smaller regions, you notice that the amount of detail you can gather is limited because of optical aberrations of the eye. These aberrations blur structures at a cellular scale. Over the last 20 years, as some of you may be aware, adaptive optics technology, developed first here at the University of Rochester, has enabled us to surpass this limitation by correcting for the aberrations in real time and enabling the view of cells at the back of the eye in vivo and non-invasively. Shown here is a near-infrared reflectance image using confocal imaging of cone and rod photoreceptors in the eye. Now, the brain occupies about 2% of the mass of the body, but consumes about 20% of its energy. And this is enabled by a very dense network of blood vessels. Now, once again, due to problems of scattering, it's difficult to see these blood vessels at a cellular scale. You can use the transparency of the eye to circumvent part of the problem, and then further use adaptive optics to zoom in on the smallest blood vessels in the eye, which is zoomed out here, captured with adaptive optics, where once again you can resolve the smallest vessels of size of about 5 microns in the eye. We now want to further build upon these advances and go beyond structural imaging alone to look at functional imaging. One way to study function is to study the changes in the structure of cells over time. And this becomes particularly relevant when thinking of blood flow. Many of you may be aware of several techniques to measure blood flow in the body. There are Doppler velocimetry-based techniques, OCT, and other phase-based approaches, which are very good at measuring blood flow in large vessels and have become the gold standard. Building upon those advances, when you zoom into the smallest vessels in the retina and other parts, limited spatial resolution and signal to noise ratio concerns limits the applicability of these approaches. Specifically, one challenge is that while it is fair to assume that blood is a fluid in larger vessels, as you go down to smaller and smaller levels, the biphasic nature of blood, which is that it's composed both of plasma and of cells, becomes important. So our goal here is to focus on the smallest network of blood vessels and image blood flow at a cellular scale. We're going to achieve this by using adaptive optics scanning light ophthalmoscopy, and I'll just give a brief introduction. First, we image the eye in vivo and use the combination of a wavefront sensor to measure the aberrations of the eye in real time, and a deformable mirror to correct for those aberrations so that a near diffraction limited spot is formed on the retina. The system works in real time in a closed loop and has a refresh rate of about 13 Hertz, which is sufficient to correct for the dynamic aberrations of the human and mouse eye. This is a more detailed layout of the system, and I'll walk you through some of the aspects here. We use near-infrared light to do our imaging, and this beam is raster scanned onto the eye. The pupil of the eye is imaged onto 4 important components of the system: the deformable mirror, which forms the corrective element, 2 scanners, the slow and fast scanner, which give you the raster scanning image, and the lenslet array of a wavefront sensor. This is a Shark-Hartman wavefront sensor used to measure the aberrations of the eye. The system works in double pass. This is because it helps us to maintain confocality. And it is composed of a series of afocal telescopes, which consists of spherical mirrors, shown here as M1 through M9. And finally, the layout you're looking at here is schematic. It is flattened in 2D. In practice, we allow the height of these mirrors to vary to compensate for third-order astigmatism and coma to give us very good optical performance at the retina. Finally, the eye itself acts as the microscope objective of the system, and the numerical aperture of the eye limits the lateral resolution that you can achieve. For the human eye, the numerical aperture is about 0.23. For the mouse eye, which is what most of the work in this thesis has been done, the numerical aperture is about 0.49, giving us a lateral resolution of near 1 micron at our imaging wavelength of 796 nanometers. And finally, the imaging light source is a superluminescent diode with a bandwidth of 17 nanometers. We intentionally do not use a laser as this helps us avoid some of the specular back reflections coming from the eye. All right. So to jump into further detail, let's look at how an image is captured. Um, the point spread function on the retina is scanned using two scanners, one scanning very fast at 15 kilohertz and the other scanning slowly at 25 hertz. The PSF is scanned across the retina, as is shown here, forms one image, and the multiple scanning in the y-axis due to the slow scanner gives you a video frame rate as is shown here. The frame rate is limited by the slow scan. So this video is playing at 25 hertz, which is the real-time frame rate at which this data is recorded. And the image you're looking at is confocal back reflected light at near infrared wavelength. For the purpose of looking at blood flow, because blood cells move very fast, the frame rate of 25 hertz is not fast enough to capture each passing blood cell in the detail that we want to. So for this purpose, we modify the scan system and freeze the slow scanner at a preferred location on a blood vessel and scan in one dimension instead of two, producing an image as shown here. This is a space-time image where each one-dimensional scan is stacked in time from left to right. By doing so, objects that move past the scan manifest as slanted lines in this image. Think of it as a motion streak, which is caused when you image an object that's moving too fast for your camera. In this particular configuration, the slope of these slanted lines gives the speed at which those objects are moving. So what you're looking at here is specularly back reflected light from individual red blood cells as they move past this scanning beam. And you can appreciate that in a very short time, several 100,000 of these cells are moving past your imaging beam. So because we know that the velocity of blood cells is given by the slope, we can always have someone go in and trace the slope of each individual blood cell. The grad student can potentially spend his entire youth doing this. However, we wanted to find a faster and more robust way. So for this, we used certain image processing techniques. We take a space-time image of moving blood cells, as is shown here, and chop it up into small regions of interest, one of which is zoomed out here. We then apply the radon transform on this image. Briefly, this means that the image is rotated across several angles, and the 1D projection of it along a specified angle is computed, as is shown here. Such a sinogram image enables you to look at linearly oriented features in such a small region of interest. Some further processing on this radon transform image gives you more insight into the orientation of structures inside this ROI. We take the variance or the standard deviation of pixel values across one axis, and the location of the peak of that variance gives you the angle at which a feature is oriented at in that image. And as mentioned before, this angle helps us calculate the velocity of that object at a cellular scale. One of the challenges of completely automated techniques is that it will always give you an answer. And sometimes, especially in biology, you need to insert logic gates to help decide if the answer is believable or how believable it is. To this end, we defined a custom signal to noise ratio metric using the peak variance divided by the mean variance of this profile, and used that to reject regions of interest of the image which did not meet a certain threshold of SNR. Shown here in this image is an overlay with cyan showing regions of interest which were rejected. And as you can appreciate, while there are certain false positives and false negatives, by and large, the automated algorithm is good at restricting its analysis to the high contrast regions of blood cells at the center of the image and rejecting the signal from areas of less confidence at the edges. Finally, this measured blood cell velocity is overlaid onto the space-time image we just saw before. You might remember that while there were several blood cells moving, we could not really discern with the human eye as to whether there was any modulation happening in those slopes or in those velocities. So we overlay the velocity in a color scale to help visually assist the interpretation of the result. And now if I play this video again with the overlaid information, you can see that there is a pulse moving through the vessel. This corresponds to the cardiac rate, the cardiac pulsation moving across this vessel. Let's look at this at a slightly larger scale. Once again, we see the raw image that was collected above with hundreds of thousands of cells moving by every second. And below, we can look at the overlaid cell velocity and appreciate the rich spatial and temporal information available there. Next, we wanted to further analyze what kind of metrics we can extract from such a method. So shown here is an analysis of the spatiotemporal dynamics of blood flow in this one vessel. You can look at the average cardiac cycle, which is shown here, and also look at the cross-sectional laminar profile and how that profile changes as a function of the cardiac cycle. You can see it beat along with the heart rate. We did some further study on what it is we can find from such a detailed look at blood flow. One of those was to look at the cross-sectional profile. We found that especially when looking at blood vessels that are this small, the flow profile, an assumption that the flow profile is parabolic is no longer valid. The profile is in fact plug-like. The black data points here show the measured flow across the cross-section of a vessel. And the red profile here shows a modeled parabolic flow with an exponent of 2. And while that model is suitable for large vessels where you can assume that blood is a fluid, when you come to smaller and smaller vessels, I have overlaid the size of a red blood cell here. That assumption breaks down and the biphasic nature of blood flow needs to be taken into account. We further quantified this and compared what happens when you actually measure the blood flow across the cross-section, which is shown in the first column here, versus if you measure just the centerline flow and then fit an assumed parabolic profile onto this. And we found that such an assumption can lead to up to a 39% error in the measured flow rate across this vessel, further emphasizing the need to measure flow at a cellular scale when talking about small blood vessels. Finally, on the theme of looking at multiple spatial levels, we tracked blood flow across different branches, starting from the largest blood vessel in the mouse retina of about 50 microns and down to smaller and smaller vessels, as shown here in this arteriolar branch. Going down to the smallest capillary and then also tracking the return flow in a vein. Now, one might ask, well, how do you know that these measurements are true? What is the error rate of your measurement? One way to look into that is to study the conservation of flow. Can also be thought of as the conservation of mass or volume by looking at a branch point as is shown here at the branch of A2, A3, and A4, and independently measuring the flow rate and then testing if conservation of flow is valid. We found that such a measurement led to an error of about 8.9%, which is one representation of the error of this approach, with the caveat that the fact that we are measuring this sequentially can sneak into some of this error. because the underlying blood flow may have changed within the few minutes that it took to go from one vessel to measuring the other. While that was in one mouse, we did an extensive population study across 20 mice to look at the normal hemodynamics in the mouse retina. Here I have plotted the velocity as a function of vessel lumen diameter. And you can see right off the bat that there is a lot of heterogeneity, that measuring diameter alone does not give you a complete picture of what is happening to the hemodynamics. If you combine velocity with diameter, you can get the volumetric flow rate, which is plotted here on the right. And this shows 133 vessels across 20 mice. Now, if you look at the capillaries, which are the smallest vessels here, which have single file flow of blood cells, you might wonder what is happening here. Is this the limit of our dynamic range? Well, not quite. If we zoom into that data, we can see that our system is able to capture small changes in flow, even at the capillary level, and looking at multiple metrics of hemodynamics, including velocity, flow rate, flux, as was measured by my colleague Andres Guevara in 2016, and also looking at the relationship between flux and flow, which helps us to further understand the variation in hematocrit at the level of these small vessels. All right, so till now, the imaging that you've been looking at is confocal backscattered light imaging, which for a long time produced a lot of insight into the functioning of the retina at a cellular scale. Over the past few years, there has been a lot of interest with several groups going beyond confocal imaging to modify the detection configuration to image multiply scattered light or forward scattered light from the retina by displacing the pinhole aperture at the detector plane to enhance the light coming from forward scatter and multiple scatter, and suppress the light coming from direct backscattered light. This leads to images, as is shown here on the right, from Chui et al. From Indiana University, where the contrast of structures which are more translucent and were previously not visible with confocal imaging alone can now be visualized using this label-free phase contrast technique. For example, you can see the edges or the vascular wall thickness as is shown here with the black arrow. And as you'll see going forward, you can see other translucent cells by this approach, which emphasizes subtle differences in the refractive index of these cells. As some of you may be aware, my colleague Andres Guevara in his optics thesis worked on enhancing the contrast of these translucent cells by not just displacing the aperture laterally in the detection plane, but also axially to improve the contrast of these cells by a factor of up to 2.7. Here he combined that approach with the fast imaging that we just saw to image a very small region of the retina at a capillary level and image individual blood cells as they passed by, once again using only near-infrared light in a non-invasive way. These bullets you see here are individual red blood cells. So I further built upon some of these advances through the following. First, I worked upon some image processing techniques to correct for the eye motion. Since we are imaging living, breathing animals, the eye is constantly moving. And in brief, I use a cross-correlation approach to correct for eye motion in these space-time images. What this enables us to do is to correct for these distortions before we start extracting any hemodynamic parameters. So for example, a raw image which looks like this, where you have a space-time image with individual red blood cells, you can see that the blood cells are shifted in space because of the natural eye motion of the anesthetized mouse. Using this post-processing technique, you can now register that image and rectify the image before doing further automated or manual analysis on this. Another question that arises is, these look like blood cells, but how do we really know that these are blood cells? To answer this, this is where the power of validating these techniques in the mouse comes through. We can use fluorescently tagged parts of the circulation to confirm that these are blood cells. Shown here, I imaged sodium fluorescein, which was intravenously injected in a mouse, and found that by simultaneously imaging in the phase contrast channel and the fluorescence channel, gaps between these putative blood cells were lit up or were positive for fluorescein, showing that these bullet-like structures in between these bright spots are indeed red blood cells, as they are not labeled by this technique. Furthermore, we were able to demonstrate visually another advantage of looking at blood cell flux using phase contrast imaging alone, as is shown here in this image. Let me point your attention to the center of the image, where you can see a very densely packed unit of red blood cells. And you can count individual ones here. So you can count 1, 2, 3. However, in the simultaneous fluorescence image, you can see that that manifests as just one solid gap, suggesting that fluorescence techniques alone may miss some of the blood cell flux, especially in tightly configured configurations, as is shown here, and therefore leading to an underreporting of cell flux in these configurations. Another advance that we worked on is looking at pulsatile flow, not just at the level of an arteriole, as I showed just now, but that pulsatile flow could be measured at the level of the smallest capillary. And this was a pretty exciting result. We achieved this by counting blood cells over time for about 80 seconds and saw that the blood cell flux was indeed pulsatile. This was exciting because the classic definition or the classic description of this in textbooks is that pulsatile flow cannot be measured at the level of capillaries and postcapillary venules. And we show here that this is only because it is a function of the sensitivity of your approach. And if you have a sensitive enough approach, like we show here with adaptive optics, you can actually tease apart these subtle changes in flow. What does this mean in the larger view of things? This means that we can potentially look at very subtle changes in blood flow, which happen in diseases like diabetes over a very long period of time. And this demonstrates the sensitivity of our technique. Finally, I worked on image processing techniques to look at the automated measurement of lumen diameter. I won't go into a lot of details about this right now, but one of the important results that came out of this was that at the capillary level also, when you plot the lumen diameter versus cell flux, notice how the lumen diameter varies just from 3 microns to 7 microns. So you're looking at a very, very small range of diameters. You can see that flux has a very, very small dependence on diameter. The diameter is a poor predictor of flux, and there is a lot of heterogeneity. And this tells us that even if you have spatial resolution to image the structure of capillaries, a measurement of the diameter of a capillary alone does not give a complete picture of the nutrient delivery happening in that capillary. And that these added metrics of flux helps us better understand what is happening at a functional level. Finally, we wanted to look at other constituents of blood in a work that I'm particularly excited to show. So once again, I show here a snapshot in time of a capillary. So this is a space-time image where time moves from left to right. And I demonstrate the imaging of the four main constituents of blood. Red blood cells, as you can see on the extreme left, followed by a gap or a plasma gap, suggesting that for a brief period of time, there were no cells flowing past the imaging beam. Quickly followed by this cell, which we now know is a white blood cell. How do we know that? because we simultaneously imaged labeled or fluorescence from labeled white blood cells using acridine orange in a mouse model. This white blood cell was immediately followed by an aggregation of red blood cells, um, and then followed by this putative platelet, which you can see here, which is of a much smaller size than the other blood cells. So what is happening here? Because this image is a snapshot in time and time moves from left to right, what's really happening is that white blood cells are known to be slightly more sticky and more rigid as they pass in single file through these capillaries, and they progressively slow down over time, which you can further visualize in this animation here, causing an aggregation or a traffic jam behind them. And this is a very special condition found only at the capillary level, because as you can imagine, as these blood cells eventually move into larger and larger size blood vessels, there is now sufficient space for the red blood cells to move around the white blood cell. Now, this was imaged in a healthy mouse, and this suggests that this is a process that happens in our eyes all the time. In fact, there is a very interesting phenomena called the blue field entoptic phenomena, which is when you stare at a blue sky, you see these bright spots moving around, which is actually caused by this group of white blood cells followed by an aggregation of red blood cells. So this is a natural process happening all the time. However, now that we can image this in the real, in the living eye, we can study what happens when diseases like diabetes or others disrupt this delicate balance between the friction that a white blood cell typically imparts to flow at the capillary level, and study what happens in such cases. So for example, when this delicate balance is disrupted, we can have disruption in flow, microaneurysms, vascular abnormalities. We also look at other characteristics of white blood cells. Shown here, we see that in some cases, the passage of a white blood cell, as is shown here in the center, confirmed here with acridine orange labeling, we found that the white blood cell leads to a temporary distension in the lumen diameter of the capillary, quantified here as a 42% increase during the passage of the white blood cell. Again, because this was done in a healthy mouse, this leads to the potential exciting application in the future where these numbers can be compared in disease. And we also observed other interesting aspects that in our label-free phase contrast imaging, the contrast of the white blood cell appears to be in some cases different from the surrounding red blood cells. We know that white blood cells have a very rough surface profile and have a very different scattering nature compared to other cells in the circulation. And this property has been used for decades now in flow cytometry, where comparisons of the forward and side scatter of light is used to classify white blood cells and subtypes of white blood cells from other cells. Given that we now see a tentative, exciting signature that these cells have in the label-free channel, this provides the possibility that future work where we no longer have to use fluorescent tags, we might be able to identify these cells just from their contrast alone. All right, so coming back to the conceptual organization of this thesis, I have shown now the imaging of blood cells using very fast imaging approaches over milliseconds to a few seconds. So let's look at the other end of this time axis. Here we want to look at immune cells, which we know from literature show very slow changes at the cellular level over long periods of time. So we are interested in a particular type of immune cell called microglia. And in the next few slides, we'll be looking at microglia in health in the mouse eye. Shown here is a commercial scanning laser ophthalmoscope image, which has a wide field of view of about 30 to 55 degrees. And we simultaneously image reflected light from the mouse retina and fluorescence in a transgenic mouse, which expresses enhanced green fluorescent protein, or GFP, in microglial cells. Overlaid on this fundus photograph is the adaptive optics image, or rather a montage of imaging fields, as is shown here in this insect. Let's zoom into that region. So you can see here that with an AO-SLO, you can image the fluorescence from these cells with a higher signal to background ratio, which is enabled by the tighter axial sectioning of the adaptive optics instrument. And you can further zoom in and see how you can image the individual processes of these immune cells. On the right here is, once again, fluorescence excited at 488 and collected at 520. And on the left here is just a teaser image of imaging a different cell in a different mouse using just label-free contrast alone. So before going into a lot of details about immune cells, let's quickly talk of some of the challenges that need to be overcome. If you look at the mouse eye in a simplistic model with the anterior optics composed of the cornea and lens, the mouse eye has a very high optical power of about 520 diopters. This compares to about only 60 diopters in the human eye. So as you can imagine, this is very high dioptric power. Because of this, imaging at different wavelengths leads to substantial chromatic aberrations, both in the axial sense, longitudinally, and in the transverse sense, laterally along the image plane. So to correct for longitudinal chromatic aberration, to ensure that we are imaging in the same plane, we independently adjust the light sources or the focus of each light source. And to correct for transverse chromatic aberration, as is represented here, we ensure that the optical axis of the imaging system and the mouse eye is roughly aligned, and use empirical approaches by imaging known anatomical structures in the eye to ensure that the TCA is minimized. All right, so we know from literature that these cells are dynamic, and they're dynamic at in a very slow sense. So we came up with the following protocol to image them across time. We had a steady beam of near-infrared light at 796 to ensure that we were imaging the same location across time, shown here across about 1.5 hours in the same mouse. And in brief intervals of 5 minutes, we turned on our fluorescence light to image the cell at these different time points. We turned off the light when we were not collecting data to minimize phototoxicity due to this light. I would emphasize that the power used for the 488 here was 53 microwatts, which is comparable to ANSI safe limits used in the human eye. So shown here is one snapshot at t=0 of a microglial cell. And this is a video, a time-lapse video of what happens to the cell over time in a healthy eye. You can see that its processes are dynamic and moving all over the place. To represent this moving image in a static 2D image, we assign a hue value to each time point, and then take the maximum intensity projection to represent movement across time in a static image, as is shown here. Although we admit that videos are way cooler to look at. So let's look at some of the characteristics of microglial cells when doing time-lapse imaging, as we just showed you. First, we saw that these cells show remodeling of its processes at multiple levels. Shown here is one example of a primary process that is retracting over about an hour of imaging, as shown on the right here. In a different cell, we show that higher order processes, including secondary and tertiary branches, are also continuously remodeling over time, as is shown in this video. And finally, by zooming in to the smallest level, we take a microglial cell here and zoom into just one branch, and then look at the smallest structures that we can see that are dynamic over time. I would point your attention to the scale bar here, which is just 4 microns. And we image that branch over about 1.5 hours, showing both that we can actually record the dynamic end protrusions of these processes. And then we can have a stable imaging over about 1.5 hours um, of time. Finally, we wanted to quantify this motility that we observed. So to do this, we built upon our previous approach of looking at space-time images to determine velocity. The difference being that the temporal scale at which we are doing this is now minutes instead of milliseconds. So we trace along the cell from the soma to the end of to the tip of the process and build these space-time images and look at displacement over time. We found that there were a variety of processes that could be observed. This analysis is done here in this plot over a population of 7 mice across 26 cells and 129 processes. We found that there were processes that were relatively stable over time. There were processes that extended and processes that retracted. On average, if you plot this on a histogram, there was a net zero sum of process extensions and retractions, showing that in a healthy eye, this activity is consistent with conservation of cell volume, which is what you'd expect. And this also provides a good validation that our measurements are correct. And we further look at the population numbers across these mice, looking at the average displacement magnitude and also the average process speed, to compare this with values found in the in vivo brain and ex vivo retina. Briefly, we found that the process velocities that we measured are comparable to that measured in the brain, and are in fact much slower than that measured in the ex vivo retina. And we comment in our paper that one reason for this is that in an ex vivo preparation, you are perturbing these immune cells and providing it with a highly toxic environment, which might change the behavior of these cells and therefore make them much more motile than they actually are in vivo. So this actually shows us the exciting possibilities of studying these cells in their native environment at a cellular scale. Finally, on the theme of imaging across time, we wanted to look at these cells over a very large temporal bandwidth, from seconds to months. So for this, we are going to use red-green merges, where 2 time points are compared, and they're red-green merged, where yellow regions of the cell show things that have not changed, and green and red regions show structures of the cell that have changed. So on an imaging interval of 40 seconds, you can see that the cell is predominantly yellow, showing that it didn't change much in that time period. However, if you increase the temporal bandwidth to 5 minutes, you can see that some changes at the level of processes can be observed. You can see a process being deleted here, shown in red, and a new process being formed here, shown in green. Imaging at an even higher bandwidth of 1.5 hours of the same cell shows even further changes along the process branches. So this is the largest we could image in vivo in the same preparation. However, because our imaging is non-invasive, because these mice are allowed to return to their cages and we can image them again at a future date, we did follow-up imaging over 1 week and 1 month at the same at the same imaging location. A question that we are often asked, and it's a very good one, is how do we know we're imaging at the same place? So for this, as we hinted upon previously, we use the near-infrared reflectance channel to ensure that. So shown here, I'm going to toggle back and forth between the t equals 0 and t equals 1 week time point. And hopefully you're convinced that it's the same location. Similarly, in a different mouse, I'm going to show the same location imaged uh, across 3 months. And you can see how we are indeed imaging the same place, the same region between two blood vessels, um, as you can see here. In this red-green merge, you can see that the yellow regions further confirm that it is the same location. So let's look at what's happening in the fluorescence channel where we track the microglial cells. You can see that over these long periods, there is some migration of these cells. And unlike the imaging times of seconds to hours, where the soma was relatively stable, we find that over these much longer time periods, there is some random locomotion of these cells in their local environment. Building upon this, we want to present the exciting result that we can image the dynamics of microglia using just the infrared channel alone, using phase contrast imaging. So shown here is the 796 nanometer axial offset imaging at the nerve fiber layer of the retina at 2 different time points. This location here was followed for 1 hour continuously. Here, I did this imaging in a transgenic mouse to help me ascertain what cell type we are looking at. So this was a CX3CR1 mouse with GFP labeling of microglia. You can see in this zoomed in version of this location that you have this cell in the label-free channel, which is positively identified as a macrophage or a microglia in the fluorescence channel. At different time points, you can see that processes that are visible in fluorescence are also visible in the label-free channel. And let's look at a much richer detail of these dynamics in the following videos. In this video here, you can see that the cell is dynamic. This is again doing continuous imaging, which is different from the time-lapse imaging you've been seeing till now, where we imaged in intervals of 5 minutes. Here, the imaging light is just on throughout. And let's zoom. First of all, before we zoom into the cell, let's appreciate how the imaging location is quite stable. You're looking at a capillary network branch here, and it's just fixed. And this is enabled using post-processing registration of this video. So let's look at the dynamics at a more detailed level. You can see in this video here that the dynamics of this microglial cell could be imaged simultaneously using both label-free and fluorescence. Process dynamics imaged in fluorescence could be imaged in the label-free channel. We wanted to quantify this. Shown here in part D is the displacement of a primary process over time, and independent measures show that the displacement is similar in the label-free and fluorescence channel, showing that in the future, label-free imaging alone could give us this important metric about microglial activity. And finally, we present the exciting prospect that there are additional details visible in the label-free cell that are missed in fluorescence. You can see that there is a subcellular flicker happening in the top right region of the soma, which one can't quite see in the fluorescence channel. And this could be putative organelle motility happening in these cells, which we could previously not image using fluorescence alone. Coming back to the time axis, here we see that we are able to image immune cells in health, starting from seconds going all the way to months. Finally, we wanted to apply these approaches in a disease model in the eye and study inflammation in the eye and what happens to both immune cells and vascular cells in inflammation. So many diseases of the eye have an associated inflammatory component, and the immune response of the retina and of the CNS at a cellular level remains poorly understood. Um, we've gained a wealth of information from techniques that study fluorescently tagged cells in animal models in peripheral tissues like the cremaster muscle, as is shown here in this image of 2-photon microscopy of the cremaster muscle. However, because labeling is required and surgery is required, we cannot do this in human beings. And more importantly, we also cannot longitudinally follow up what is happening to these cells, because many of these approaches offer just one time point one time point of imaging because of the invasive nature. So the goal of this part of the thesis is to optimize recent advances in adaptive optics to image label-free immune cell dynamics and the vascular response in inflammation. So for this, we use a model of inflammation called EIU, or endotoxin-induced uveitis, where we inject lipopolysaccharide, which is effectively dead bacterial cell wall, into the vitreous of the eye in a mouse. And this induces an inflammatory response, which is very acute, and the time course of which has been well characterized. This work that I show here was done in collaboration with Colin Chu from the University of Bristol and NEI, where he worked with us in Rochester over a summer to do the study. His work in the past had shown that looking at optical coherence tomography images of the eye when it is healthy and after it has been given this disease or this acute inflammation, you can see that in an axial cross section where you are looking at the retina at the bottom and the vitreous on the top, there is this infiltrate or debris that you can see here with these punctate white dots shown by the yellow arrow. However, this is just one snapshot in time, and the question remained of what this debris actually was. And this was a perfect problem for adaptive optics to solve. So here, we imaged a mouse when it was healthy using adaptive optics phase contrast imaging with 796 nanometers. And we gave it this acute infection and then imaged it again after 3 days. We see at the same location that there is clearly something that has happened to the retina. There is a lot of debris around this vessel here. And we asked ourselves what this debris was. Are these cells? Are they living? Are they dead? And where did they come from? So to answer that question, we realized that we were perhaps imaging a little too late in the game, which was 3 days post inflammation. And we decided to again induce this disease model in a different mouse and image a little bit earlier. When we did that and we focused in on a blood vessel, we found that in this real-time video here, that there are these cells that appear to be sticking to the wall of a vein and moving or rolling very slowly across the vascular wall. So this was done in a mouse just 6 hours post the induction of inflammation. What you're looking at here is the first step in a cascade of events which helps the body to fight infection. And you're looking at the recruitment of white blood cells here. When there is an infection, there are chemical signals that are released in the tissue, which signal to the white blood cell and the endothelium cells along the vascular wall to express selectins and integrins, which instruct the cell to start sticking to the wall of the vessel. These cells first start rolling across the vessel wall, eventually come to a stop, and then extravasate from the vessel through a process known as transendothelial migration. So this provided us the very exciting result that using label-free imaging alone, you could image this first step in this cascade, And as you will see soon, several other steps as well. The advantage of doing this first in the mouse before translating to a human being is that we can confirm the type of cells that we're looking at using genetically modified mouse here, or using other fluorescent tags. Here we injected a label which specifically tags a subtype of white blood cell called the neutrophil using Ly6G Alexa Fleur labeling. And in this simultaneously captured video, you can see that these rolling cells across the edge of the vessel correspond to the brightly fluorescent cells here at the bottom, showing us that these are indeed neutrophils. We then went back to our debris field around the vessel, which we saw at 3 days post the induction of disease, and now performed slow imaging or time-lapse imaging. So in this sped up video, you can see that those cells are indeed alive. These are white blood cells and other immune cells, which have extravasated from the vessel and are now fighting disease outside of it. Every cell here has its own story. Each is doing its own thing, possibly following invisible markers of cytokine and chemokine labeling. And I'm going to draw in your attention to a few special processes that are happening. So let's first focus in on this red circle here, where you will see, if you stare long enough, that there is a cell that appears to extravasate from the vessel and go outside into the tissue to fight infection. In a different example, in this green region shown here, you can see the opposite happening, where a different white blood cell, which was outside in the tissue, ate up debris, did its job, and has now decided to re-enter the circulation and go back into the blood vessel. And these are just two examples, but as you can see clearly, there is a lot happening here and a lot of exciting work that is going to be done in this in the future. We imaged further dynamics of these cells. We looked at subcellular dynamics happening inside resident macrophages or myeloid cells. As you can see here, using phase contrast imaging, you can image which is activity happening inside the soma, which is possibly organelle or lysosomic motility. And this is very exciting, as it was clearly not visible in the fluorescence imaging alone, and offers new ways to study the activity of these cells in an unperturbed preparation inside. And this work was a co-first author effort with Colin Chu, our collaborator, which was published end of last year in Eli. We wanted to, because this approach was non-invasive, we wanted to look at other metrics that we have become very good at obtaining from the retina. So in conjunction with immune cells and their motility in inflammation, we wanted to apply the single cell blood flow measurement that we started off this presentation with. So remember again, that blood flow is proportional to the cross-sectional area of the vessel multiplied by the average velocity of blood cells. I imaged label-free a vein and artery in this disease model, where I imaged at baseline and then at 6 hours, 1 day, 3 days, and 10 days post-infection at the same location. You can see that the vein appears to dilate at the 24-hour time point and then resolve back to its baseline state at the 10-day time point. This dilation and then resolution was similar to the inflammatory component seen in the eye, where the number of recruited blood cells in the retina also increased up till the 24-hour time point and then resolved by about 10 days. And this disease model has been characterized to show that it's an acute model which peaks and then resolves, which enables us to image the entire timeline or pipeline of the immune response. As you can see in the bottom row, an arteriole, which was located right next to this venule, showed comparatively very little change in its lumen diameter. Now, we know from the first half of this presentation that diameter alone is a poor predictor of hemodynamics, and it needs to be looked at in conjunction with blood velocity. So using the space-time velocity imaging, we saw that in the arteriole, um, as the inflammation progresses, the blood velocity increases dramatically. What you're looking at here are individual space-time images. You can see the pulses, which are the cardiac cycle pulses, and red is high velocity and blue is low velocity. So you can see that the average blood velocity has increased substantially in the artery by the 24-hour time point, and then it resolves towards baseline by 10 days. Conversely, in the vein, in a relative sense, there are less changes happening in the blood velocity. So this provides us an interesting duality of these two processes. So this was shown in just one mouse. So we studied blood flow in a population of 6 mice to further tease apart the biological variability. And what we found was that indeed, on average, the veins dilate in response to inflammation. The arterioles are relatively unchanged in terms of diameter. However, on the velocity axis, the opposite happens. In the veins, there are less changes, while in arteries, there is a substantial increase in blood velocity. Combined, when you look at the volumetric blood flow, we find that the change is in tandem, which is kind of what you would expect, given there are no leakages happening in the retina, that in a complete system, the amount of blood flowing through the artery is then collected in vein. So this gave us the very interesting result that first, blood flow increases substantially, almost up to 80% in inflammation, and then resolves toward baseline, and that the mechanism of this increase is very different between arterioles and venules. And one also remembers that veins are the site at which immune cells choose to start rolling and then extravasate. And this might have something to do with the fact of why it's just the veins that dilate, and that the arteries, which have a much stiffer vascular wall, are able to compensate for that increase in blood flow by allowing a larger blood velocity without allowing its diameter to change. And further study should be able to tease this apart. Finally, we present this exciting result that some of these approaches have already been translated to the human being. Shown here is blood velocity measured in a human eye using a different AO-SLO designed just for the human eye. And you can see that the pulsatile rate is much slower than in the anesthetized mouse. While in the mouse, you have, say, a heart rate of 220 beats per minute. You have the more familiar heart rate of about 60 beats per minute recorded in the human retina in a small blood vessel. So coming back to where we started off at, which is looking at techniques, both optical and otherwise, on two axes of space and time. You can appreciate how some of the approaches we've shown here occupy an impressive space, especially on the time axis. The fact that you can follow the same cell over very long periods due to the non-invasive nature of our imaging. One of the limitations of adaptive optics has always been its small field of view. And as you can see clearly here, we're very good at imaging cellular changes but in small fields. And the hope is that these approaches, while certainly not meant to replace other techniques, that these approaches can provide or complement other techniques to start studying the interrelationships between cells, layers, and the entire central nervous system. In terms of future directions, there are several, and I'll point out a few. First,
just to give you a quick heads up, we're at about 15 minutes past, and I'm wondering if you can go over the future directions quickly, just to give us enough time for the closed door session as well.
Sure, sounds good. Yeah. So I'll do this very quick, just in like another 2 minutes. So label-free imaging provides translational potential, and the use of these measures of blood cell speed and process motility, we can perform a cellular scale study of diseases which impact the vascular and immune systems. We can do a non-invasive study of inflammation in the CNS, which has previously required the need for surgery or cranial windows in animal models, and do longitudinal studies in diseases that move very slowly across the human body, like diabetes and others. And also the fact that right now we've only shown the simultaneous study of, say, leukocyte dynamics and blood flow, but there are other metrics that we can follow up, like cell flux and others. And we really only scratched the surface in terms of label-free imaging of white blood cells. And we can further distinguish between subtypes of cells by looking a little more closely at the different scattering properties. This work resulted in the following publications, patents, and first author conference presentations. And finally, I'd really like to thank first my advisor, without whom this work would not have been possible, and who's always been very supportive, just like you see in this picture here. The ARIA lab, which have spent several hours helping me fine-tune these presentations and my research, and also my committee, my chair, the co-authors of my papers, and the departments of optics and CVS, and my parents, family, and friends. Thank you.
Thank you very much, Aby. I did want to ask that the committee please save their questions for the closed door, especially given the time. But if there are any pressing questions from the open session, please do ask them now. You can use the chat feature or just unmute yourself and ask Aby directly.
Hi, Aby. Could I ask a question? I thought it was a fantastic presentation and really an amazing body of work. So thanks for that. I was wondering, it was really difficult to do these time-lapse imaging sessions. And I was wondering if you'd done anything particular to stabilize the axial position, and if you had any thoughts about whether the axial PSF for the fluorescence and the reflectance might be different, and whether that made an impact on the differences you saw between the microglia images.
Sure. Yeah. Yeah, that's a very important point. Yeah, I think certainly there could be some differences in the axial PSF between the two imaging wavelengths. You would expect, at least from a confocal theory of light, that the axial PSF is slightly larger for the infrared light source. However, we have found that while that's true for confocal imaging, in phase contrast imaging, we find for some reason the sensitivity to axial position it's actually much easier to refine that in the near infrared channel as compared to the confocal fluorescence channel. We try our best to make sure that we're at the same axial plane by switching from fluorescence to visible light reflectance using the same wavelength, so a green laser light source, and doing confocal imaging at the green wavelength to make sure that we are roughly matched axially. The error to within which we can do this is the diameter of a capillary, which is about 5 microns. That is the smallest biological unit we use to make sure we're at the same place. So hopefully that gives a sense of to what degree you can make sure you're at the same place. But certainly that is a continuing source of worry that some of the differences could be because of axial misalignments.
Okay, great, thanks.
I'd like to limit to just one last question, if there are any.
I'll ask one. Hey, Aby. Hello from Seattle. Great
So,
talk.
thank
Uh,
you.
I just wanted to ask about transverse chromatic aberration in the mouse eye. And you had a slide talking about it. So, you know, I remember from our first year optics class that, the transverse chromatic aberration scales with the longitudinal chromatic aberration, and that if you knock down all LCA, the TCA should disappear, right? But in your case, you're focusing the infrared and the visible light at the same place, yet some transverse chromatic aberration remains, and you have some approaches to correct for it. So if you could just elaborate on that, that'd be great.
Sure. Yeah, that's a great question. As you correctly mentioned, correcting for one certainly reduces the magnitude of the other to a large extent. What I found while actually performing the imaging was that there is still some small uncorrected TCA after I corrected for the LCA. And this, I assumed, was because of displacement of the mouse eye itself relative to the optical axis of the system. And what I found was if I, in real time, did the simple task of adjusting the XY position of the head of the mouse itself, that I could see in the images that there was a lateral shear, a lateral misalignment that was being corrected just by positioning the eye pupil more closely to the optical axis of the system. So as I just described to Juliet, the procedure to calibrate this was to image using visible reflectance and to look at reflectance images of a capillary network to make sure that the displacements or the TCA is minimized. And again, this was a little bit of eyeballing, but when you're looking at this in a large screen, it helps you to minimize that to within the width of a capillary. Thank you.
Great. In the spirit of time, I'd like to thank everyone for your attendance for this open portion of the defense. I'd like to ask the committee members, Aby's committee members, to please use the link that you were provided for the closed door session. Aby, I'll ask for you to sign off. I will send you a message when you're ready to rejoin the room. And please all take a break and we'll reconvene in just a few minutes. Thank you so much.
Thank you. All right, Jesse, I'm going to sign off.
Sounds good, Aby. Thank you so much. And I will send you a text via WhatsApp, okay?
Sounds good.